Hi, my name is Ariel Munitz, and I wish to present here with the highlights of our recently published study entitled Distinct Roles for TSLP and IL-33 in Experimental EOE. Eosinophilic esophagitis, or in short EOE, is a chronic food antigen-induced type 2 associated allergic and immune-mediated disease. It is clinically displayed by dysphagia, vomiting, chest and abdominal pain, food impaction, and often failure to thrive. Histopathologically, you will find eosinophilic infiltration and often eosinophilic abscesses in the epithelial layer of patients. You will also observe basal cell hypoplasia and epithelial thickening, as well as elongation of the papillae, dilated intercellular spaces, and if chronic and untreated, you will also see a very fibrotic diseases. Using an experimental model of experimental EOE, we have recently shown that IL-13 is a key mediator of EOE. It is upstream of eotaxins which drive in the eosinophilia observed in the disease. It is responsible for epithelial cell proliferation and decreased barrier function. It induces a strong fibrotic response and induces angiogenesis. But the upstream regulators of IL-13 in EOE are relatively unknown. In the recent study which I'm presenting now, we hypothesized that TSLP and IL-33 are upstream of IL-13. We were strongly interested in TSLP and IL-33 since these are epithelial cell-derived cytokines which are likely secreted and upregulated at an early stage of the disease. We have first shown that IL-33 expression is increased in experimental EOE in mice, and we confirmed this data using single-cell RNA sequencing databases showing that similar to mice, also in humans, epithelial cells are a good source for IL-33. Similar to IL-33, TSLP expression is also induced in the mouse system in the experimental model, and also in the human data, you will see a very strong signal coming for TSLP expression from epithelial cells. And this raises a question. If both TSLP and IL-33 are induced, which one of them drives disease? Do they both work together? Does any one of them have a significant and distinct function? Or do they have distinct roles? What is the division of labor between these two? In order to answer this question, we went to knockout models. Using IL-33 deficient mice, we induced experimental EOE in these mice, and we could show that epithelial thickness and Ki-67 epithelial cell proliferation was hardly affected in the absence of IL-33. We did observe a slight decrease in the accumulation of eosinophils in the esophagus, but basically IL-33 deficient mice were pretty much as sick as their wild-type counterparts. So does TSLP have a role in experimental EOE? To answer this question, we assessed the roles of TSLP using TSLP receptor knockouts. As you can clearly see, eosinophilic infiltration in the TSLP receptor knockouts was completely abrogated. Similarly, basal cell hyperplasia and epithelial thickness, as well as lamina propria thickness, was completely reduced in the absence of TSLP receptor. We confirmed these findings using anti-TSLP treatment by treating the mice with anti-TSLP antibodies, and could also show that this type of treatment reduced disease in mice. Finally, we have shown that TSLP has a key role in EOE pathogenesis by showing RNA sequencing data that TSLP is upstream of IL-13 signaling in the esophagus. Finally, I would like to thank Anish De Silva, who carried this work nearly single-handedly, to all of our lab members and to our collaborators, and thank you for your time.